Just confirm me, am I audible to you? Just confirm me, am I audible and the PPT is visible? Okay, fine. Let's start today's class. See, in today's class, we are going to see about same thing, physiology and reproduction in mammals and bonds, chemical bonds, and also some of the other topics. Okay, okay, fine. So let's start with a bond type. Okay, what are all the types of bonds and where it is getting used and some of its examples. Okay, so the first bond will be ionic bond. And see, when it comes to ionic bond, for example, NaCl, we can separate this bond by Na plus Cl minus. Transfer of electrons. Okay, one is gaining and one is losing the electrons. This transfer of electrons from one atom to another, we are calling them as the ionic bond, right? See, formed by the transfer of electrons from one atom to the another, resulting in the attraction of oppositely charged ions. So this is what an ionic bond is. So example will be the sodium chloride and potassium iodide. We can separate this potassium iodide as K plus and I minus. One is gaining the electrons and one is losing the electrons. Okay, when it lost the electrons, it gains a positive charge like that. Okay, now what is covalent bond? See, you should know that here the sharing of electrons will be happen between the atoms to complete their valence shells. See, we know that I hope you all know the basic chemistry, which means this K shell, L shell, M shell like that. And in first shell, 2 atoms can be filled, and in second, 8 will be filled, and the third 18 will be filled like that. Okay, so to complete their valence shell, the electrons will be shared. Okay, so that is what a covalent bond. Here, the electrons will be shared. But in ionic bonds, here the electrons will be transferred. Okay, and examples will be the hydrogen, oxygen, and water. Now, what is polar covalent bond? In polar covalent bond, this is the type of the bond. Here, the electrons are getting shared unequally between the atoms, and they are creating the partial charges. Okay, like hydrogen fluoride and water. Okay, so this is what a polar covalent bond. When we dissolve in this particular, if we dissolve a particular compound, this can be considered as a solvent. So that is why we are calling them as the polar covalent bond. Okay, now what is metallic bond? Definitely, as the name suggests, it will be found in the metals. Okay, now here the electrons are getting delocalized, which means what? They are changing their position and they are moving freely. And here, and the electrons will be moving freely among the positively charged metal ions. That's why metallic bond. Copper, iron, gold, these are all the metals, and the bond formed between these metals is called as the metallic bonds. Now, hydrogen bond. This hydrogen bond is considered as a weak bond, and this bond will be happen between the hydrogen atom and an highly electronegative atom like the oxygen, nitrogen, and the fluorine. These are all considered as the electronegative atom. So this will be between the hydrogen bond and will be between the electronegative atom. So for example, water molecules and between the DNA strands. We know that the nitrogenous bases are getting connected through this hydrogen bond. Between the adenine and thymine, we have 2 hydrogen bonds, and between guanine and cytosine, we have 3 hydrogen bonds. Okay, so and you should know that that will be the weak bond. Now, van der Waals force, again, this will be the weaker attraction, and that will be formed between the molecules or parts of the molecules that arise from the transient fluctuation in the electron density. For example, here that will be a non-polar molecule, such as hydrocarbons. This is not a polar compound. This is a non-polar molecules. No. So this is the basic understanding of the bond type. Okay, these are all the list of bonds. So next will be this will be the overview of the biomolecules. See that this organic compound, we have carbohydrates, lipids, proteins, and nucleic acids. In carbohydrates, we know that there will be a general formula CH2O of N. This will be the general molecular formula of the carbohydrate. Okay, and what is said to be as the carbohydrate? They are considered as the polyhydroxyaldehydes or polyhydroxyketones. We know that carbohydrates is also called as the polyhydroxy. Poly means many, hydroxy means OH group, aldehydes means CHO. This is what polyhydroxyaldehydes or polyhydroxyketones. Ketone means CO group and aldehyde means CHO group. So this will be the thing you have to remember. This in case of carbohydrates. We know what is lipids and proteins and the nucleic acids. Now coming to the subunits of carbohydrates. See, these carbohydrates are considered as sugars. That will be the sugar molecule, and we have a monosaccharide, disaccharide, and polysaccharide. Okay, and everything will be the sugars like starches and cellulose. Everything will be coming under the carbohydrates. Now fats. When it comes to the lipid, when we break them, see, whatever it is, whether it will be the disaccharide or polysaccharide, when we hydrolyze those saccharides, those polysaccharides and disaccharides, we can get a simpler sugars. We can get a monosaccharides. Like that, when we dissolve this lipid, we can get glycerol and fatty acids. That will be the basic subunit of the lipids. Okay, so that we know that will be the fats and oils, and we have two different types of fats. One will be the saturated fatty acid and another will be the unsaturated fatty acid. And these lipids will have a major portion when it comes to the cell membranes. When it comes to the plasma membrane, lipids secures a major position. Right? Now, proteins. We know that proteins will be the building blocks of the matter. Am I audible to you? Everything is perfect, right? Is there any problem? Okay, I'm sorry. Okay, fine. See, this lipids will be having secure a major position in the plasma membrane. Okay, we know that what will be the model present put forth by the Singer and Nicholson in case of plasma membrane. So, lipid will plays a major role in case of a cellular membrane. Keep in mind that. And proteins. It is made up of an amino acid. See how many amino acids are present in the world, or we can say in the earth. How many amino acids are present? Okay. Is there only 20 amino acids present in the earth? The earth. Oh, is it 21? Oh, 64. Why it is 64? See, we have 300 amino acids. Okay. But the most commonly existing amino acids will be the 20 amino acids. Okay. Right? Don't forget this. The most commonly existing amino acids will be the 20, but we have a 300 amino acids. Okay, fine. Now, and we should know that one amino acid is getting attached with the other amino acid through this particular bond called the peptide bond. That bond will be formed as a result of a condensation reaction. What does mean by condensation reaction? Through the elimination of audio disturbances. Okay. Is it clear now? Is it clear now? Is it clear now? Okay, wait.
Recording in progress.
Now? Okay, I hope. Is it clear now? Now. Is it okay
Is it
now?
okay now?
Okay, it is
Okay,
happening.
it is happening.
Okay, check now.
Okay, check now.
Is it okay now?
Is it okay now?
Nah.
Oh, okay now.
Okay.
Okay.
Okay. Now,
Okay. Now,
is there any problem now?
is there any problem now?
Okay, fine.
Okay, fine.
Okay, if you find any
Okay, if you
disturbances
find any
again,
disturbances
just let me
again,
know. Okay.
just let me know. Okay.
From where I have to
From
start?
where I have to start?
From where I
From
have
where
to
I have
start?
to start?
Just tell
Just
me.
tell me.
Oh my
Oh
God.
my God.
What is it? Okay. I think now it is clear, right? Okay, I'm sorry. Okay, sorry for the disturbances. Okay, where I have to start? Where I have to start? From amino acids? Quick, quick, we have less more number of time. Amino acids, okay. See, when it comes to the amino acids, we do not have only this 20 amino acids. You should know that we have a lot of amino acids, approximately 300, but the most commonly existing amino acids will be 20. Okay, so see, when it comes to the nucleic and one amino acid will be connecting to the other amino acid, which by which bond, that will be an important thing, that will be the peptide bond. And this peptide bond will be formed as a result of the condensation reaction. And you should know that what is mean by condensation reaction. That is nothing but the bond will be formed by the elimination of water. Okay, that is what a peptide bond. And nucleic acid, we know that we have two different types of nucleic acids. One is deoxyribonucleic acid and another one is a ribonucleic acid. Okay, now you should know that what is deoxyribonucleic acid and ribonucleic acid and all the basic things, you know that. And these nucleic acids contains a nucleotides. Okay, and nucleotides, and these nucleotides have a bases and sugar, nitrogenous base, pentose sugar, and nucleotides. These are comprisingly called as the nucleic acids. Okay, so this is what an overview of the biomolecules. And now coming back to the structure of a protein. Okay, see, we have a different structure of a protein: primary structure, secondary structure, tertiary structure, and quaternary structure. See, you should know this primary structure will be the linear sequence of the amino acids. There will be no branch will be seen in case of this particular primary structure of a protein. It is just a sequence of amino acids. It is linked through a peptide bond in a polypeptide chain. Okay, this is what a primary structure is. And you should know what will be the example. This will be the sequence of acid in a protein. But in the secondary structure, we can find the foldings. Okay, we can find the folding pattern, an arrangement of polypeptide backbones stabilized by the hydrogen bonds. Okay, and see this here, you should think, you should know what are all the bonds are involving in this particular structure. See, that will be an important thing. See, in case of a secondary structure, you should know the hydrogen bond will be involving here, right? And this secondary structure have an alpha helix and beta pleated sheet and beta turn. So okay, so because of this structure, it results in the folding pattern. Okay, this is what a secondary structure, and you should know that the bond involved here will be the hydrogen bond. Now tertiary structure, here the overall three-dimensional structure, this will be the three-dimensional structure of a single protein molecule determined by interactions between the amino acid side chains. Side chains is nothing but the R groups, okay? And here the structure, tertiary proteins will be seen in the globular proteins and fibrous proteins, okay? And it is highly functional in nature. And quaternary structure, here the arrangement of multiple protein subunits and the interactions between them to form a functional protein complex. This is also a most functional protein. We can find the hemoglobin, DNA polymerase and ribosomes have this particular quaternary structure. Okay, so I want you to revise the PPT which was issued by the sir. What are the things you have to focus there will be the, what are all the bonds involved in the quaternary structure and in the tertiary structure and in the secondary structure. The bonds involved in these structure plays an important role. Okay, now that's all about the protein structure, and you should know some of the basic basic information about the Ramachandran plot here. See, we have it at two axes, the x-axis and the y-axis. Okay, and the x-axis represents the torsional angle, that is psi, okay, in degrees, and y-axis represents the torsional angle phi. Okay, it is phi and it is psi. So this will be the axis. And what are all the data points? Here, the data points represent the amino acid residue. Okay, what will be the exact amino acid residue present in this particular protein? That is what a data points, and the coordinates of each point correspond to the phi and psi torsional angles of the residue. Through this, we can find what will be the structure present in this particular protein. See, in case it will be the, see here we can find the positive values, here we can find the negative values. Here we can find the negative values and here we can find the positive values. So you should know that here will be the positive and this will be the negative. Here, this will be the negative and positive. Here, this will be the negative and positive. Here will be the positive and positive. Okay, this will be the negative and negative. See, there will be no protein residue will be present here. If it is present in the minus area, then we should know that the alpha helix right-handed structure will be present in this particular protein. If it is present in the plus area, definitely that will be the left-handed alpha helix. If the residue is present in the minus plus, definitely the structure will be the beta pleated sheet. Okay, this is how you should know that. This is how we can find the residue, okay, of that particular protein structure. Now, what will be the regions? We have a different regions in case of this Ramachandran plot: favored region, allowed region, and outlier region. Outlier region. In favored region, see, it is located in the central area of the plot. Here, these areas represent the combination of psi and phi angles that are energetically favorable and commonly observed in the protein structure, psi and the phi area. Okay. And allowed regions will be the surrounding area of the favored region. The favored region is getting surrounded by the allowed region, and here these allowed regions have a torsional angles that are permissible but less common. Here in favored regions, the protein structures are commonly observed proteins are in this particular favored region, but here less common. This allowed region will be the less common, and outlier region will be the, here it will be the periphery of the plot. Periphery of the plot means surface of the plot. And here the residues in this region have a psi and phi angles. It will be rarely observed in the protein structure. So commonly observed region will be the favored region, and less common will be observed in the allowed region, and rarely observed region will be the outlier region. And here, if it is found in this particular region, then we can find that will be the structural abnormalities and there will be the error in that particular protein. Okay, now colors. In case of a favored region, it will be depicted in the green color, and it will be the most energetically favorable conformations if it is depicted in the green color. And in the allowed region, it will be shown in the yellow or the orange. to denote it will be acceptable but less preferred conformations. An outlier region will be marked in the red color. Why it is in the red color means it will be the danger symbol. Okay, that will be whether there will be any problem. Is there any problem? It will be depicted in the outlier region in the red color. Now, what will be the analysis we can get from this particular Ramachandran plot? See, by examining the distribution of the data points on the plot, the researchers can assess the quality and validity of the protein structure. We can find the qualitative analysis. We can say like that. Okay, and here, the region with high density of the data points indicate the well-folded and stable regions of the protein. If it is a well-folded, then the protein structure will be either quaternary or tertiary. Okay, we can find the structure of the protein as well. Now, the outliers may suggest the errors in the protein model. Is there any error or a mistake in that protein structure? That will be mentioned in that will be depicted in the outlier region. Is there any if it is there any abnormalities, then it will be shown in the outlier region. Now, what are all the interpretations we can interpret in this particular plot? It provides the insights into the local conformational preferences of the amino acid residues within the protein structure. Is there any conformational change? Is there any structural change? It can be depicted in the Ramachandran plot, and it is also useful to understand the stereochemical constraints that govern the protein and stability. See, if the protein wants to be stable and if the protein has to fold properly, definitely there will be the involvement of a bond. Okay, so we can also analyze what are all the bonds, that is stereochemical constraints will be there in the protein, which ensures the protein stability and folding. We can come up into a conclusion. Now, see, deviations from this favored region and allowed region will indicate the potential areas of refinement or further investigation. See, if it is not shown in the favored and allowed region, we have to analyze the protein once again. So this is what an interpretation we can do it in the Ramachandran plot. Okay, now what are all the applications in this particular plot? What are all the uses? Once again, quality of the protein structure governed by the experimental proteins, qualitative analysis, just like the X-ray crystallography and NMR spectroscopy. And see, we can find the protein model. Okay, by using the protein model, we can do many discoveries. For that, it will be necessary. And if there is any mutation, what are all the effects of mutation? What are all the ligand will bind to that particular protein? What are all the receptors present in that? And what are all the post-translational modifications can be undergone by this particular protein? Everything will be analyzed by this plot. Now, what are all the limitations? Limitations mean what are all the disadvantages? Which are all the things which cannot do under this Ramachandran plot? This is based on the empirical observations. It is not capture all the possible conformations, which means what? It is just the practical observations. It is just an experimental observations. It is not practically in use. Okay. Now, it does not account for dynamic changes in the protein structure or influence the environmental factors. And here it needs an expertise in structural biology and protein chemistry. Not a layman will do this particular experiment. Okay, only an expert can analyze this particular plot correctly. Okay, now coming to the reproduction. Okay, let's solve some of the questions, and after that we'll go to the reproduction in mammals. Which type of bond involves the sharing of electron pairs between the atoms? Sharing of electron. Here I didn't say anything about a transfer of electron pairs. Transfer of electrons. Here the question will be there as the sharing of electron. Okay, don't get confused. Now give me a correct answer. That will be the covalent bond. See, in case of a transfer of, in case of ionic bond, the electrons will be shared. electron will be transferred. Here, the electrons will be sharing, okay, to balance the valence shell. That is what a covalent bond is, okay. And you should know this covalent bond will be the strongest bond. And this will be the reproduction. Which of the following bond is formed between the metal and the non-metal? The questions come from this particular topics to the exam will be very low, okay. But I want you to know all the basic information in your syllabus. So that is why I included this. Between a metal and non-metal. 15 minutes more so that answer quickly. We'll finish it off the reproduction in mammals also. That will be the ionic bond. See, it will be between the metals in case of a metallic bond. Ionic bond will be formed between a metal and non-metal. Only then there will be a transfer of electrons from one atom to the another atom that will be held together by these forces called the electrostatic forces. This will be a definition, and just remember this. Okay, that will be formed between the metal and the non-metal. Okay, now which bond is characterized by attraction between partially positive hydrogen atoms and partially negative atoms in the other molecules? Don't think it will be the chemistry part, it will be the biochemistry. Okay. Okay, see, hydrogen bond. Okay, as I said, I have explained you this point. See, this hydrogen bond is occurred between the molecules with the hydrogen atoms. Definitely, if there is an involvement of hydrogen atoms, there will be the involvement of this particular bond also. This bond, this hydrogen bond will be formed between the hydrogen molecule and will be the between the electronegative atoms like oxygen, nitrogen, and fluorine. Okay, so it is a weak bond, but crucial for many biological processes such as DNA and protein folding. You should know that. Okay, now which type of bond do atoms share electrons unevenly due to differences in electronegativity? I think nobody liked this part. I think nobody is answering. Everybody liked biology, but not chemistry. See, sharing of electrons unevenly due to the differences in electronegativity is covalent bond. Okay, see, there will be a difference in electronegativity between the bonded atom. The electron here is not sharing equally. Just remember, the sharing of electrons is always deal with the covalent bond. Okay, and which bond is responsible for high thermal and electrical conductivity observed in metals? Okay, if you find difficulty in these topics, just read the notes which have been attached in the first 2 to 3 slides. That is okay. That is sufficient, right? And also try to read all these questions. That is more than sufficient. That will be obviously the metallic bonds. Okay, and which biomolecule serves as a primary energy storage molecule in cells?
primary energy storage molecule in cells. Quick, we have only 10 minutes. Yeah, carbohydrate. See, everybody like biology but not chemistry, right? Which biomolecule is composed of nucleotides and functions in the storage and transmission of genetic information? Oh, okay. Why it is? Okay, answer. Here, this biomolecule composed of nucleotides and functions in storage and transmission of genetic information. And that will be none other than our DNA. Why it will be a protein? Okay, right. I don't think that this DNA is not a biomolecule. Everything in our body is a biomolecule. Okay, and which biomolecule serves as enzymes, hormones, antibodies, structural components in cell? That will be the end product of our DNA. The DNA has to be converted to that. Only then it will be considered as a building block of our body. That will be a protein. Okay, protein is everything. Okay, now the ultimate goal of DNA that it has to code any protein. Okay, now which biomolecule is composed of a long chain of amino acids linked by a peptide bond? Yeah, that will be protein. Okay, if it is a peptide bond, that will be obviously protein. Which biomolecule functions in the synthesis of proteins based on the genetic information encoded in DNA? Which biomolecules functions in the synthesis of proteins based on the genetic information encoded in DNA? Just think and answer for this question. Don't confuse with the protein here and RNA and anything. The DNA has to replicate, and that will be converted into RNA, and only then it will be converted into a protein. Okay, this will be the central dogma of the molecular biology. So that will be RNA. Okay, now which of the following is not a function of a protein in living organisms? This will be not a function of protein. Enzyme catalysis, energy storage, structural support, transport of molecules. That will be energy storage. See, transport of molecules. We have many transporter. Okay, to make the passage for all the ions, molecules, and all. Okay, so everything will be the protein. Only thing it is not do will be the energy storage. That will be related to the carbohydrate and lipids, but not proteins. Okay. Now, which level of protein structure is determined by the sequence of amino acids? Here, just we can say only the sequence of amino acids and not any other. And that will be the primary structure, no doubt in that. And which type of protein structure is characterized by the folding and twisting of the polypeptide chain into the alpha helices and beta sheets? Alpha helix and beta pleated sheet. And that will be the secondary structure. Okay. And what will be the bond involved in this particular structure will be the hydrogen bond. Okay. Now, which level of protein structure involves the overall three-dimensional arrangement of secondary structural elements and other motifs within a single polypeptide chain? Very good. That will be the tertiary structure. Okay. Now, which level of protein structure involves the association of multiple polypeptide chains to form a functional protein complex? Here, there will be the association of multiple polypeptide chains to form a functional protein complex. And that will be the quaternary structure. Okay. So, this is what everything, bonds, biomolecules, and protein structure, a Ramachandran plot. Okay. Everything. And if we start reproduction in mammals, and we don't have enough time for that. So, we'll stop up to this. And for you, tomorrow's homework will be the glycolysis. Okay, this full line. Glycolysis, carbohydrate metabolism, and TCA, tricarboxylic acid citric acid cycle, and their biomedical importance, pentose phosphate pathway, PPP pathway, and gluconeogenesis. Okay. And redox potentials, mitochondrial electron transport chain, oxidative phosphorylation. Okay. Okay. If lipids, okay, we'll finish it. Okay. Carbohydrates. See, carbohydrates, proteins, and lipids. Carbohydrate chemistry and metabolism, protein chemistry and metabolism, lipid chemistry and metabolism. Everything will be covered tomorrow. And also, besides, you should read this cycles, glycolysis, TCA cycle. Okay, and the entire thing will be carbohydrate, lipid and proteins, both their chemistry and metabolism. Everything will be covered tomorrow. Okay, fine. Is there any doubts? Based on the time, we'll see the next things like mitochondrial electron transport chain. I think everything will be completed in tomorrow's class. Okay, anything else? After that, we'll start evolution, developmental biology and all. Okay, any doubts? Don't worry, no topics will be pending at the end of the day. Everything will be cleared. And also, if you need any topics to teach, that will also be taken into a consideration. Okay. Anything else?